ions, a lot of them in there, if you have looked at them before. And check this. These dots are representing mineral ions, and this structure here is actually representing the cell membrane, in this case of the cells of the root hairs. And then you can look at this side, you're having small dots. Again, these dots are representing mineral ions. Now, if you are so much observant, it should be noted that on one end, if I can call this end of the cell membrane, let me call it A, and this side let me call it B. On one end called A, you're having a high concentration of mineral ions, but on end B, there is a low concentration of the mineral ions. So that means if diffusion is to occur, whether facilitated or the normal diffusion, that means the concentration has to be different across the membrane, and it has to be down the concentration gradient. Okay, that is from a region of higher concentration of the molecules to a region of low concentration of the molecules. So if the concentration of a particular ion in the soil solution is greater than its concentration in the root hairs, that is, now this side would be the outer, and this side of the membrane will be the inner part of the cell, of the root hair cell. Now the mineral ion is going to enter the root hair by diffusion, and this process you don't need any energy, and that's why we call it a passive process. It's a passive process, but sometimes it could be active. It could be active, whereby if the concentration, look at this side, you're having a lot of molecules inside the root hair cell. Okay, let me write the word here, inside. This is inside root hair cell. Root hair cell. This is inside the root hair cell. And then let me get some of these people. They are bringing chaos. Just a moment. I mute these people who are approved. Okay. So this side, inside, you see that there's a lot of mineral ions inside the root hair cell, and this is out maybe in the soil water solution. This is out in the soil water solution. Okay, this is soil water. This is the soil water solution. Now, if you're having in the soil water solution, there are less mineral ions compared to inside. So diffusion cannot occur, but much as these minerals could be still needed, so such minerals are going to be taken up by active transport, which means that energy is going to be involved to actively take them, that is against the concentration gradient. There are two words involved here. There is, you could talk about down a concentration gradient, or against a concentration gradient. Against. Now, if it is against, it means that molecules are being diffused from where they are at a low concentration to where they are at a high concentration. But for the case of when you want to use the word down the concentration gradient, it means molecules are moving from where they are at a high concentration to where they are at a low concentration. Now remember, with facilitated diffusion, there are some channel proteins through which these ions can pass through to move to the other side. Okay, Hillary says, sorry teacher, I have just joined, but what are we at? We're in biology. This is senior 5 biology. So if you're not in senior 5, you better leave the lesson because you'll get lost. Then the mechanism of mineral ion uptake can be can be illustrated using a graph. And for most of this time, such numbers actually in paper 2, as well as even paper 1, because such a graph can be brought in paper 1 as well, in section B. Now, when you're looking at this graph, for any graph actually, for any graph in biology. Whenever you're given a graph, first read the words, the title words, because they sometimes have some clues of how you can tackle that graph. So here it says that the figure that is 22.16, this is the figure, shows this is the relationship between the rate of respiration and mineral uptake. You can see they're showing a relationship between the rate of respiration and mineral uptake. Now, the second thing after reading this is you read the words on the vertical axis as well as on the horizontal axis. Now, the words on the vertical axis here, you have rate, and this is supposed to be T. This is respiration, but this rate is being measured as the amount of oxygen taken up. Take note of that the rate of respiration is being measured as the amount of oxygen taken up. That means when there is a lot of oxygen being taken up, it means that the rate of respiration is high. And I think you know the definition for respiration. Now, it should be noted, very many people tend to confuse, and even up to this level, by the way, in A level, but I think by A level, we assume that you already know. Some people tend to think that respiration is taking in air and taking it out. No, that should never even ever cross your mind. But rather, respiration is going to involve breakdown of glucose or sugars to release energy, that is in form of ATP. But this breakdown occurs in presence of either oxygen or absence. If it occurs in presence of oxygen, it is called aerobic respiration. If it occurs in absence of oxygen, then it is called anaerobic respiration. But of course, in most cases, anaerobic respiration yields less energy in different organisms, especially plants and animals. So most of the time, the kind of respiration that occurs is the one that involves use of oxygen to break down the sugars or glucose to release energy in form of ATP. Okay. Now, this is trying to explain the mechanism of mineral iron. Now, look at this graph. It is on the horizontal axis, it is having time in hours. And here, from the start here, you can see that you are having distilled water, okay? And the rate of respiration was a little bit moderate. You can see here, this line is showing that the rate of respiration is a little bit moderate and constant. You can see, moderate and constant. Okay, and this is when you are having distilled water. So, but then at this point, after 2 hours, potassium chloride was added at this point after 2 hours. And when you add in potassium chloride, check what is happening to the rate of respiration. It is increasing rapidly. You can see it is increasing rapidly. That is with additional potassium chloride solution. So what do you think, or why do you think the rate of respiration increased in this case when you added potassium chloride solution to this culture? Let's say, I'm assuming this was happening in, let's say, cells, a culture of cells. A culture, when you're culturing cells, that means you're growing cells somewhere. So why do you think these cells are going to take up the rate of respiration in these cells is going to increase rapidly when you've added potassium chloride to the solution where they are? Who can think about that? What do you think? You can raise your hand and you tell me. I don't know whether you guys are getting me. Are you getting my question? Yes, Alfred. Yes, Alfred.
Yes, sir.
Yes, please.
Are you hearing me? Yeah.
Yes, I'm hearing you. You can go on and
I think if you add the potassium chloride, it is going to add more concentrated. So outside the root cell, there is going to be more concentrated than inside. So what
Okay.
is going to, so it's going to move inside. Yeah.
Okay, thank you for that contribution. Yeah, thank you. But the contribution have not had anything to do with respiration. Should I say sorry, teacher? What? As I say yes, teacher, we are getting it. Yes, that's why I needed you to give your submission on this. Why is it that um, here the rate of respiration in the cells was constant, but then when potassium chloride was added to the solution where these cells are, it increased rapidly? Why do you think the rate of respiration increased? Because this is how you're supposed to be able to interpret these kind of graphs. If you can't interpret such a graph, then it means you've not yet gotten the concept. So who can tell us why? Why? I'm saying we are having very many students here, but I'm not seeing anyone raising the answer. Can anyone say something about it? Okay, I think everyone has failed to interpret this. Yes, Arthur?
Are you hearing me?
Yes, I can hear you, Arthur.
Sure, what I think, when we added potassium chloride, it acted like a catalyst in the reaction. So it increased the rate of respiration.
Thank you
Yes.
for that contribution. You are saying that it acted as a catalyst. Remember, with catalyst, they increase the rate of reaction of certain processes. Daphne is saying to pardon the question. The question is very clear in this graph, is that here, when these cells were in a culture, okay, where they were put, being placed or preserved in distilled water, and the rate of respiration was just constant. But in that solution where these cells were, when potassium chloride was added to that solution where these cells were, the rate of respiration increased rapidly. You can see that it increased rapidly. And I'm saying that in potassium chloride solution, why is it that the rate of respiration increases? That is the rate of respiration. You see, you guys, you should always take note of any word on the graph, because it is telling you that measured as the amount of oxygen taken up. Why is it that the amount of oxygen taken up increases rapidly? That is when you've added potassium chloride solution. I think there now I've softened it. Yes, or Michael.
Teacher.
Yes.
When potassium chloride is added to the solution, it makes concentration of water inside the cell higher than outside the cell. Hence it makes the cell to begin taking in water actively. That is what makes the rate of respiration increase.
Oh, Odeke.
Yes.
Do you think water can be taken up by active transport?
Yes.
Sure. What happened? What happened to osmosis?
The mineral salts, not the water.
The mineral salts. There. Now, can you modify your answer again?
Addition of potassium chloride increases the concentration of mineral salts inside the cell than outside the cell, hence making the cell.
Wait. Why do you say that inside the cell, yet the potassium chloride has been added to the outside?
Teacher, I'm saying inside. I don't know why.
Okay, let us first get on Arthur and then we see. Yes, Arthur?
Teacher,
Yes.
what I think, when potassium chloride was added, the concentration gradient also increased. So it enabled the water to diffuse into, outside, no? Because potassium chloride was added outside. So the concentration gradient increased, increasing the rate of respiration.
Okay. Thank you, Arthur, for that. Now, what I've seen, most of you guys, you've not yet learned how to clearly kind of relate concepts here in this case. Because remember, I had just talked about active transport, okay? Where under active transport, I said that if the concentration of a particular ion in the soil solution is lower than in the root hair cell, like in this case whereby I use this scenario in the corner, that you could have a lot of minerals inside the cell as compared to the outside, but you need this inside. That thing can be taken up against the concentration gradient, okay? And here we use energy. Okay, to simplify it for you, this figure here shows that when salts are added to plants growing in water, this concept of growing plants in water is called hydroponics, because it has been done in UK, whereby scientists discover that you don't need soil to grow plants or to grow crops. You don't actually need soil, but rather you need, you could have water, and then you add there all the mineral salts that you will ever need, and the plants are going to grow and give you good yields even without soil. So this is the concept where they got it from. So this figure shows that when salts are added to plants growing in water, the respiration rate of the plants increases, presumably in order to provide the energy for mineral salts absorption. You get it? Because for you to absorb, if you are a cell and you are in water and you need a certain nutrient, okay, you're going to take it up. But sometimes you may find that inside already you're having a lot of mineral salts, but you could take it up by active transport. And active transport means that energy must be available. But that energy can only be available after respiration, that is breaking down sugars to release energy in the form of ATP. So once energy is released, that is, in order to release that energy under aerobic respiration, so you must take up, if you are a cell, you must take up a lot of oxygen. And that's why here, the rate of respiration that is measured as the amount of oxygen shoots up very fast. It increases rapidly because you have to break down these sugars to release energy, which energy is going to be used in mineral salt absorption. Okay. Now the addition of the, okay, I didn't want to bring in that at this moment. Sometimes when you add a respiratory inhibitor, then active uptake of mineral salts may not occur. And then the absorption is going to be left to only by passive means, that is facilitated diffusion or the normal diffusion. Okay. So I hope you guys have gotten that. So with passive absorption, if the concentration of a mineral in the soil solution is greater than its concentration in the root hair, the mineral may enter the root hair by diffusion. That is down the concentration gradient, and that kind of process does energy. Okay, but then also by active transport, if the concentration of a mineral in the soil solution is less than that in the root hair, that is the thing can like passive absorption can't occur and the mineral is needed, then it may be taken up by active transport. And by the way, most minerals are absorbed in this way, and the process is normally very selective because it is passing it is happening in a selectively permeable membrane, and it will need energy in form of ATP in order to absorb this, which energy is provided for by the process of respiration. Now we are having another small graph here, which is saying that the effect of respiratory inhibitor, that is in this case using cyanide, on mineral uptake. Cyanide is a poison which has been used, by the way, in like it was even one time used by the Nazis during the Second World War. In the Second World War, if you were a spy and you were caught, these guys were given cyanide poisons. So in case you didn't want to be tortured by your enemies, that people would just swallow cyanide tablets and they would die instantly. Okay. So so the effect here, we're looking at the effect of respiratory inhibitor cyanide on mineral uptake. Still, I told you that when you're given a graph like this, always read these words, then read the words on the vertical as well as on the horizontal axis, then read the words on the graph. And then you begin relating concepts, because normally these concepts which are related, and even in your explanations, you should always relate concepts. Now here we are having the rate of potassium uptake. Okay, this is rate of potassium uptake increasing upward. And then on the horizontal axis, we are having time in hours. We are having rate of potassium uptake in the solution of potassium chloride. So it was high. So potassium uptake, why? Because it is within, let's say, the potassium chloride solution. So it was very high, probably because you may guess why. But then, after about 1 hour and let's say 50 minutes, 50 something minutes here, potassium cyanide was added. Remember, potassium cyanide is a respiratory inhibitor. Yes, Simwe, you have a question?
Yes, teacher, I would like to try describing that graph, if you don't mind.
Please go on.
Okay. I'm going to do as if it's paper 2, number 1.
Okay, no problem.
Okay. Initially, at the beginning of the experiment, from 0 to 1 and, let me see, 1 and 50 minutes, yeah, the rate of potassium uptake was high. Then at 1 hour and 40 minutes, the rate of potassium uptake dropped rapidly. And then from 1 hours and 40 minutes to, let me see, to 2 hours and 20 minutes, the rate of potassium uptake reduced gradually. And from 2 hours and 20 minutes to the end of the experiment, the rate of potassium uptake almost leveled off. This is because
thank you. Okay, thank you. Yeah, for that description. Now there are some words which I have noted from your description, and I think you can see them on the graph. And I will begin with the first one, which is saying initially. I think you are somewhere where people are shouting. You may pause, you may mute. Okay. So I was still telling you that I've described well, but let us modify your description a little bit. Now, when you use the word initially means this point at the start. Here, initially means this point at the start. All, yes. So when you say initially, that means this trend is just positioned there. It's not moving. So you cannot say that initially the rate of potassium uptake is, you said it is from Simanya from 0 to 1 and 40 minutes somewhere there. So when you want to use the initially, just describe that point alone and then put a semicolon. Then you can now begin moving the train. For example, you can say initially the rate of potassium uptake is high, full stop, or you can use a semicolon. That's the verb. It is high initially, okay? Then afterwards you start from, let's say, 0 hours to 1 hour and 47 let's say minutes. Then the rate of potassium uptake is constant. You see, it is constant. Then from 1 hour and 47 minutes to let's say 2 hours and 30 minutes, the rate of potassium uptake decreases gradually. Okay, this is rapid. From let's say 1 hour and 47 minutes to let's say 2 hours. Yes, here rate of potassium uptake decreases rapidly. Then from 2 hours to 2 hours and 30 minutes, rate of potassium uptake decreases gradually. Then from 2 hours and 30 minutes to 3 hours, rate of potassium uptake is almost constant. Okay, but then remember, it doesn't zero. So normally it's not good to use this word drop because these words are relative. Let us and as well as reduced, let us learn to use the word decrease. But then you tell us the extent to which the thing is decreasing. Does it increase rapidly? Does it decrease rapidly? Does it decrease gradually? Okay, so let us not use these words which are normally used in slang, dropped. Okay, then leveled, let us use almost constant. So I thank you for trying to bring that out well for that description, and I hope each one of you has been able to pick out something on how we can describe it. Now, if we can, let me first get my pen here. Okay, my pen is there. So if you can you give me a reason as to why at this point of 1 hour, let's say 46 minutes, after adding potassium cyanide, the rate of this rate of potassium uptake decreased rapidly? Who can give us the reason? Yes, Asimo.
Okay, in my understanding, I think by that time, you see they're applying potassium cyanide, and it is a respiratory poison or inhibitor. So it actually stops respiration. So there's no ATP to active transport. The whole mineral uptake is dependent on diffusion, which is actually very slow, and therefore the drop in the decrease in the mineral uptake, the potassium uptake.
Okay, then why is it that it does not decrease to 0?
Okay, I can't really be certain. Like, wait, it doesn't decrease to 0 because there is diffusion taking place, and the uptake of mineral salts is not only dependent on active transport, it is also dependent on diffusion. So inhibiting one doesn't really totally stop the whole uptake of mineral salts, and diffusion does not require use of energy.
Okay, thank you very much. And I want to assume that each one of you has understood from Samuel's description. Okay, so because these graphs, the moment you do not understand these graphs, by the way, it becomes very hard for you to get paper 2. Number 1, especially number 1, because that number 1 normally has about 40 marks. Now, For some of you have forgotten, when we are relating this thing, we are relating the mineral ions in the soil water solution, as well as the inside of the root hair cells, because the absorption is supposed to occur from the soil water solution to inside the root hair cells. So when we are relating these concepts, have this in your mind. You should have always have this picture in your mind. So MRF is saying that I think it's because the potassium cyanide is a respiratory inhibitor, stopping the formation of energy in form of ATP. And which ATP? Yes, MRF, this is a good reasoning. And remember, this energy in form of ATP is the one which is used. Oh, let me mute someone here. Okay. So it is the one which is used under active transport when these root hairs are taking up these mineral salts. And I think you can see this is a taproot, and in taproot it could have those root hairs, the root hair cells, and then mineral salts are being taken up. But of course, when you're describing, apart from understanding those two concepts of, let's say, diffusion as well as active transport, you should always remember the pathway taken by these mineral salts until they reach up the plant. Never forget that kind of pathway. Maybe this Namayanja Diana. So never forget such a pathway when you're describing these two processes. So after the mineral salts have been absorbed, they move towards the vesicular tissue. Remember, I said that the vesicular tissue in this case is the xylem. So they move by that process of diffusion as well as active transport through the simplest pathway. And I think, remember, I talked of the simplest pathway, which is the pathway, remember, that is going through the protoplast. For some of you have forgotten the meaning of the word protoplast. Means protoplast. These are the protoplast. These are the living contents within the cell, and the living contents within the cell are normally surrounded by the cell membrane, and they are in the cytoplasm. So that's why this is taken through the cytoplasm, because that's where the living contents of the cells are, all just through the protoplasm or protoplasts. Okay, protoplasts. So the endodermis is, remember, known to concentrate the mineral ions before they are passed into the vesicular tissue. What special thing is known about the endodermis? Remember, this was the route from the epidermal side to the cortical cell side to the endodermis here. Now, this endodermis has what we call a Casparian strip within. The Casparian strip is suberized. That is, it has a lot of suberin in it, so it is impermeable. Its cell walls are impermeable to water. So the water is rather diverted. That is, the water that has been moving through the cell walls via the apoplast pathway, it is diverted to move via the simplest pathway, that is in the living contents of the cell, also call them the cytoplasm. Okay, so once it has been concentrated, so it is going to enter into the vesicular tissue, which is the xylem. And remember, I described the xylem last time. The xylem is made up of two tissues, which are water conducting tissue, that is the vessel element as well as the tracheids. And here you can see the vessel. Okay, you can add here vessel element. That is if you want it. Vessel element, but even if you just write vessel. So from the vessel element and tracheid, the mineral ions are transported along with water up the stem from the roots into the leaves by the transpiration stream. Remember the cohesion tension theory, which we talked about last time. Now, the mineral ions finally carried into the photosynthetic cells of the leaves and other growing parts of the plant, that is from the xylem vessels and the tracheids, where they are used in building up amino acids and proteins. Okay, you guys eat these plants and you get proteins, you eat beans, you eat what? So how do these proteins, how are they built up? It's after some of these minerals have been out from the soil up to the plant. Okay. Okay. So now, if you have understood this concept, I told you this paper, this question, these guys. So remember, I told you that this is a paper that was brought last year. Okay, let me first put this down here down. Yeah, so UNEB, that is Uganda National Examination Board, Uganda Advanced Certificate of Education 2020. Okay, these are the guys that you are going to follow to sit. Paper 2, its code is P530/2. This is the question which was brought. This is the graph which they gave them. It is the very question which was also examined. Actually, it was so much similar to the one which came in Wakishamo. And what was it saying? Of course, wheat is growing not so much in Uganda, maybe in the Canadian prairies, if you study geography. So in an experiment, cells were obtained from the root of wheat seedlings, were studied in a culture media of varying oxygen concentration, full stop. The effect of oxygen concentrations on the uptake of potassium ions in the root cell sap and the consumption of sugar by the root cell sap were investigated, and the results presented as shown in this figure. What does the figure say? On the vertical axis, you can see here concentration of potassium ions in root cell. It is here measured in millicurate per liter, And then on the horizontal axis, you're having the percentage of oxygen in the culture solution. So that means these are percentages: 0%, 10%, 20%, 30%. You see? And then on this side, because this was having two vertical axes, it is having concentration of sugar in root cell sap. So here you can see whether the sugar concentration is decreasing or increasing. Here you can see whether concentration of potassium ions in the root cell sap is increasing or decreasing, and you can relate it always related to the percentage of oxygen in the culture solution. What I've just done is what I want you guys to always do when you are tackling such numbers of paper 2, because it is a problem very many people fail to interpret them, and they end up writing nothing and they lose 40 marks. You can imagine. Year in, year out, when in marking, you find that very many people fail to get any mark on this number. Okay. So study the figure and answer the questions that follow. So that's how you're supposed to already study. Of course, I skipped number 1, part A, because it was about description. And I am assuming that each one of you knows how to describe anything here. And part B was saying, explain the effect of increasing the percentage of oxygen in the culture solution on the concentration of 1, sugar in the root cell sap. And this was 11 marks. So 11 marks. And then of increasing the percentage of oxygen in the culture solution on the concentration of potassium ions in the root cell sap. This was 8 marks. This was 8 marks. Okay. So if you guys have understood what I've just taught you, because that's the information that you needed if you had sat UNEB last year. So can you be able to explain the effect of increasing the percentage of oxygen in the culture solution on the concentration of sugar in the root cell sap? Can you all we may first skip this and maybe we do this. But also you go for it. Why not? Because we've just discussed it. Some of you could be passive learners and you are just there listening. What about the rest? I'm seeing many people, Maclean, Praise, or Adongo, or someone called Galaxy, Wakuma Joshua. All people are here. Rita, Okwalinga, Joe, Odeke, Emily, Margaret, Namaganda, Brivian, Mildred, Miriam, Imbusa, Kaganda. So all you guys are here and you've been following me. Proceed. So what's up? What do you have to say on this part B? Yes. How would we go for this? Yes, Asimwe.
Teacher, I can try describing, but I'm not really sure it can earn me 11 marks.
No, don't describe. I want you to explain.
Yes, okay, explaining, like, okay, whatever explanation I'm giving, I'm not really sure it is going to give me 11 marks. But I'm just going to try.
Okay, go on. Go on.
Okay, let's see. The effect. The okay. From 0%, from 0% of oxygen in the culture solution to 25% of oxygen in the culture solution, the concentration of sugar in the root sap decreased rapidly. This is because let me see. Yeah, active transport is taking place, and there is need for the sugar. The glucose is being oxidized in the presence of oxygen to give out ATP that is necessary to facilitate active transport for uptake of potassium ions in the root cell sap. Yeah, maybe. I'm not really sure.
Yeah, thank you very much. Yeah, you have some points there. You could have scored like about 5. That was a very good attempt. That was a very good attempt. What about the rest? What do you say? What do you say? Now, if this number had come in your name, does it mean you guys would get 0? Does it mean you guys would get 0? Okay, let us first pause this somewhere. Now let us go to part 2. Let us go to part 2. Part 2 says that explain the effect of increasing the percentage of oxygen in the culture solution on the concentration of potassium ions in the root cell sap. Can I have someone to explain the effect of increasing the percentage of oxygen in the culture solution on the concentration of potassium ions in the root cell sap? What is it? I wouldn't want to give you guys direct answers yet. We have just done this. We have done this. So if you cannot use this knowledge that we have just looked at here, and then it means you guys were not paying attention. We're having only one student in class. Okay, yes, Asimwe, can you say something about it?
Okay.
Yes.
From 0% of oxygen in the culture solution to 36% of oxygen in the culture solution, the rate of potassium, the concentration of potassium ions in the cell sap increased rapidly. reaching its maximum.
Mm.
This is because increasing in the oxygen concentration increased the rate of oxidation of respiration that yielded more energy to yielded more ATP to facilitate active diffusion. Active transport, which led to the rapid increase of uptake of potassium ions in the root cell sap.
Okay. Yeah. Thank you very much. I think you are one of the good students who have understood this concept.
So
and
many
then
marks.
There was you could have gotten about 6 out of 8.
Thank you.
Because I've been noting the different marking points in my head. And then another question would be, what would happen to the concentration of potassium ions in the root cell sap if in another experiment potassium cyanide was added in small quantities into the culture solution at 20% oxygen? Are you seeing that? So that means you had to have understood okay, no, Amy is saying increase in the oxygen percentage produces energy in form of ATP, which is required for active transport from the culture solution. Okay, so that means you must have understood what I've just talked about in order for you to get the concept. Now let us have a brief look at the answers which we have. So, for example, increase in oxygen within the culture solution from 0 to about 36.5% leads to a decrease in sugar concentration. Now, why does the examiner use leads to a decrease? Because you are explaining the effect of, if you don't bring in the aspect of leading to all causes this, it means you're not describing an effect of some within culture solution from 0 to 36 point leads to decrease in sugar concentration, and then you give us reasons due to increase in aerobic respiration where sugar is used as a substrate. All these are, whenever you see a semicolon, that's a full mark, to form ATP for active pumping of potassium ions into root hair cells. Further increase in oxygen concentration from that 36.5 to 60% has no effect on sugar concentration. I think this is what we have looked at, and these are the different answers. Because of time, I'm seeing we are looking about 1 minute, actually 2 minutes. Then for part 2, increase in oxygen concentration from 0 to 25% leads to increase in oxygen concentration of potassium ions in the root cell sap due to aerobic respiration, or you could use breakdown of sugar, forming a lot of ATP. I think that's why whenever you are talking about ATP, I could note that that's a full mark. Pumping of potassium ions in the root cell, that's another full mark. Are you seeing that? Further increase in oxygen from 25 to 60 has no effect due to accumulation of carbon dioxide that lowers the pH. Accumulation of carbon dioxide that lowers pH. So respiratory enzymes inhibited and active uptake of potassium decreases. So we are left with 1 minute. Yes, Asimwa, you have a question?
Yes, teacher. On this one of further increase in oxygen concentration. Okay, they brought in the idea of inhibition by the pH. How about since the enzymes working, we bring in also the idea of the active sites are all already occupied, so further increase in oxygen, like oxygen is no longer a limiting factor. How would it be if I looked at it in that direction?
In that direction, it wouldn't work out well, because oxygen in that case is not the substrate, which can be which can affect due to maybe occupation of the active sites, because this thing is happening. The substrate in this case are the sugars. So oxygen is just going to, you can't bring it out like that. Okay. I don't know whether you've got it.
Yes, teacher.
Yeah. So you can't use it as a like filling the active sites and so on and so forth. Now, because of time, of course, all results in 1 hour, and it's time. So we shall meet same time, same next week on Monday. Unless you have any question, goodbye and enjoy yourselves. Okay, Daphne says she has learned a lot. Thank you. Bye.